these video notes are going to go over the process of mitosis. Mitosis is a process of cell division, and the result of mitosis is two identical daughter cells. And daughter cells are just how we refer to new cells. So daughter cells are the cells that we're going to make here at the end. So here are two new cells. And then that means that our original cell is known as the parent cell. Just to go over a brief overview of what's happening, mitosis happens when we're making copies of what are called somatic cells. Those are the cells of your body. So for humans, those are your regular body cells that have 46 chromosomes each. And when we want to make new body cells that also have 46 chromosomes each. So mitosis happens when we are growing, when we need to repair tissue, or we need to replace tissue. Mitosis is also the way that some asexual, meaning not sexual, organisms reproduce. So they basically make clones of themselves. So there are certain plants where you can cut them and they'll reproduce and grow a whole new tree from that cutting. And that's because they only use mitosis. And so they're able to make new organisms, but those new organisms are clones, basically, of the originals because they're just genetically identical to them. During mitosis, there is the possibility of mutations when DNA is being copied, but otherwise our expectation is that the daughter cells are identical to one another and that they are identical to the original parent cell. So to go over a brief overview of what is happening, so I drew the parent cell two different ways. So technically, the parent cell has a whole bunch of DNA on the inside, and it will look like this, but that gets a little bit confusing when we're drawing. So I also drew the parent cell like this, so that you could clearly see that there are 4 chromosomes in this organism. Now, I know we're not used to seeing chromosomes like this, but technically, this contains all of the genetic information that you need in only this one chromosome that consists of only one chromatid. So when we have 46 chromosomes, if they were to all line up and be condensed and all wound super tight, it would be 46 of these bars. Now, during the first stage of prophase, DNA replicates itself. And so that's when we get these sort of X shapes that we're more familiar with seeing. So here is still one chromosome. So we still consider this X shape to be one chromosome, but it's made up of 2 chromatids. And technically these 2 chromatids are identical. So this chromatid and this chromatid look exactly the same. So this is where the copy of the DNA is. So now we've got 4 chromosomes, but they're all copied. So they're all attached to their own copy. So they're all wound up nice and tight. The nuclear envelope is just starting to break down, and we've got some microtubules that are going to start to extend. And you don't have to know much about the microtubules, but just if you see pictures, that's what they are. Then what happens is we take those 4 chromosomes that are made up of those 2 identical chromatids, and they line up on what's called the metaphase plate. So that's just the middle of the cell. And it's those little microtubules coming from these are called centrosomes, but you don't have to know that. They're going to attach each to a side, each to a chromatid. So each of the microtubules are attaching to one side of that chromosome. And then during anaphase, what's going to happen is that the 2 chromatids that were once attached to each other are now going to separate. So this chromosome and this chromosome, or this chromatid and this chromatid, are identical to one another. And so they're going to separate to opposite poles. That's what's going to happen to all of those chromosomes. And then during telophase, new nuclear envelopes are starting to form. The process of cytokinesis starts to happen. And that's just the pinching here in the middle until we have 2 separate cells. And if you notice, these cells in the nucleus have 1, 2, 3, 4 chromosomes. 1, 2, 3, 4 chromosomes. So each of these new daughter cells each have 4 chromosomes inside. This one has 4 chromosomes inside, and then no longer tightly wound because they only tightly wind during mitosis. And this cell is identical to its sister cell, and both of these cells are identical to that very original parent cell. So they each have 4 chromosomes, and they are identical. So that's what happens in humans during mitosis as well. We have 46 chromosomes. Those 46 chromosomes replicate the DNA, so we get 46 of these types of chromosomes, ones that are made up of 2 chromatids each. Those 46 chromosomes all line up on the metaphase plate. Then we get 46 of these guys splitting to opposite poles. And then what we get during telophase is 46 chromosomes in this left pole and 46 chromosomes here on the right pole. And then once it totally splits, we've got 2 new cells. 46 chromosomes in one of the new cells, 46 chromosomes in the other new cell, and those both are identical to the original parent cell that also had 46 chromosomes.